Hey, welcome to the Bioinformatics Chat. Today I have two guests, Amrit Dhar from the University of Washington and Fred Hutchinson Cancer Research Center, and Kristian Davidsen from Fred Hutchinson Cancer Research Center. Guys, welcome to the podcast.
Thanks for having me. Thanks for having me. Yeah, thanks for having
for us. Yes.
Yeah. So it's a funny thing. This is the second podcast in a row that I have with the guests from Seattle, and I guess third podcast in total. So there must be many good and interesting people there.
I heard the last one. Yeah, you had one with Amy Willis.
Amy Willis, yeah.
Yeah. That was interesting, yeah.
And so you guys wrote a paper, co-authored a paper titled Predicting B-cell receptor substitution profiles using public repertoire data. And you wrote a software that's called SPURF to do the inference of B-cell receptor substitution profiles. But let's start with you. Tell us, what do you do now, how you got there? Kristian, maybe let's start with you.
Mm-hmm. I'm currently sitting as a programmer / analyst in the Madsen group at Fred Hutch Cancer Research Center. And my story about, so I am Danish, and I am from Copenhagen, Denmark. And that's where I did my undergrad. And throughout undergrad, I was visiting some different countries, and I got, excited about going to the US. And at some point, halfway through my master's, I found out that I wanted to make a project about B cells and B cell receptor evolution. And so, as it turned out, I ended up in Seattle with Erik Madsen, because he was doing work that I found interesting, and we had some similar interests. So I ended up contacting him, and we got to know each other, and I went to Seattle, and I stayed there since. So I've now been in Seattle for 1.5 years, and I will be starting my PhD this summer. And so in that 1.5 years, I've been working on a handful of different projects, and one of them with Amrit, who's now the co-author of this paper together with me, and it's been great fun.
Cool. Amrit, what about you?
Yeah, I'm originally from California, and so I did my undergrad in California, and just very I was interested in just pure statistics and a little bit of mathematics here and there. I had no interest in biology at all. I thought it was very just memorization of terms and very just not a real logical field at all. And when I came to do my PhD here, I got really into phylogenetics, which is the study of kind of evolutionary relationships among different species, different just sequences, and somehow and then a little here here and there, I somehow got to be part of Erik Madsen's lab and kind of focused kind of phylogenetic inference problems with respect to B cells. So, I'm certainly not, I didn't study, kind of B cell dynamics from a biology perspective, but I'm certainly picking up, all the information as I go.
That makes sense. So we'll be talking a lot about, B cells and B cell receptors and clonal families. And I don't want to assume that all the people who are listening to this know all that stuff. And certainly my knowledge is limited. So can you give a brief introduction to these concepts?
Yeah, I think that responsibility falls on me. So, so there's many terms. This is immunology, and there's there's just so many terms and abbreviations, and it can be extremely hard for, I think, even a trained immunologist to follow them. But there's some basic concepts for B cells. And so B cells are like an elementary part of the adaptive immune system, and so you could sort of divide the adaptive immune system into two parts. There's the B cell compartment and there's the T cell compartment. And these are the two compartments that can dynamically adapt, and those are the compartments you can activate to elicit a vaccine response, so you can get protective immunity, and it's also what will fight off infections and all sorts of good things. But P cells are one compartment, and that's what we are. And I should say that they are really intertwined, so they're connected in many ways. So you can't really segregate them out like that, but that's a simplistic view of looking at it. So P cells are the cells that create what is commonly known as antibodies. And I think most people have some idea about what antibodies are and what they do. And antibodies, just to recapitulate that, is small proteins, small Y-shaped protein molecules that can bind and then neutralize pathogens. And so that might be viruses, that might be bacteria, it might be proteins that can be toxic, something like that. But the point is that they can bind to all sorts of things, all sorts of biological surfaces. So that might be proteins, it might be glycans, it might be, pretty much anything. And so you might then ask how is that's one of the fundamental questions in immunology when it started out, that people were doing these experiments where they took a mouse and then they basically injected some kind of pathogen into the mouse, and then they could observe that they got very sick. First time they got this injection, and the second time they were re-exposed, then they were not getting very sick. So of course they could see that the mouse had adapted to something. And as it turns out, one of those responses is the B cells. So they're actually undergoing small, like microscale evolution to bind and neutralize whatever you get injected into your body. And in like at the cell level, what happens is that the B cells are getting produced continuously. There is a lot of B cells that are produced in the bone marrow all the time. And those B cells are what is referred to as naive B cells. And these naive B cells have a receptor. That's the B cell receptor. And the B cell receptor is the surface-bound form of the antibody. So now, all these B cells that have different receptors, they have like a DNA recombination mechanism that makes them extremely diverse, so that whenever there's a new recombination, then you can be fairly certain that it's a recombination which has not been observed before. So there's a continuous creation of new and diverse B cell receptors. So that's like a pool of receptors that can potentially bind something, that they can all potentially bind those pathogens. But just because you have one or two cells that bind something, that doesn't make up for an effective immune system, because those two cells won't really be able to do very much. Right? So you need one thing at least. You need expansion of those cells that react towards the immunogen. And as it turns out, you also need something else. You need something that makes them bind better. Because these naive receptors that are supposed to bind and recognize pathogens, they might bind very weakly, and that's simply not enough to have an effective neutralization of the pathogen. So this is where, so long behold, now we come to to the process which we have studied intensively in the Madsen group, which is affinity maturation. So this is the concept where you have some protein which is immunogenic. So this is foreign protein. You have an immune response which is elicited against this. So it all starts out with one or a handful of B cells, naive B cells. They recognize the antigen, but they recognize it quite weakly. And then, because they recognize it, and because they're naive B cells, then they can undergo a process called affinity maturation. And that is a process that happens in the lymph nodes, and there's some nodule-like structures that are formed called germinal centers. And in those germinal centers, there is a selection process, and there's a proliferation mutation process. And it's divided into two zones. It's called a light zone and a dark zone. And in the light zone, there's a selection for high or good antigen binders. So B cells that have mutated to produce a better receptor. And in the dark zone of this germinal center, there is a different process which is cell division, vast cell differentiation, not differentiation, sorry, division. And then there's a mutation process which is special for affinity maturation, and that's called somatic hypermutation. And the reason for that is that this receptor is not very long, and if you are to make an efficient and fast evolutionary process, you gotta introduce some mutations, and just like the normal baseline somatic mutation rate is so low that you would basically never get anywhere. So there's a special process for boosting this mutation rate to around 1 millionfold of the normal somatic mutation rate. Okay, so that's the basis for affinity maturation. You have selection, and you have proliferation and mutations. And after some time, we're talking like 3-4 weeks or so, you actually have accumulated a good number of mutations in these receptors. Around 5 to 10% of the nucleotides will have a mutation, and usually you observe that the affinity, so the strength with which the B cell receptor is binding the antigen is around 1,000-fold higher, maybe even 10,000-fold higher. So there's like a substantial improvement in these receptors. In this process, there is like quite a vast number of cells. We're talking like 10,000 cells that are inside this germinal center, and it's actually that process, that microevolutionary process, that has been the center of attention for many projects in the Madsen group, including this project. So if you boil away all the fat from what I've just said, we are actually just trying to make predictions of the substitution process, the underlying substitution process of this affinity maturation. So I hope that makes somewhat sense.
This makes sense, yeah. And also specifically, so you're not looking at the mutation process for a specific antigen, right? But you are interested in the question of like which nucleotides and which amino acids we can mutate at all, right? Which ones does it make sense to mutate?
Absolutely. Well, I would say first of all, there's a massive problem within the community of like analyzing B cell receptor sequences that we hardly know what they bind. So that's a recurring problem again and again, that we can sequence a lot of these because these can be sequenced with some with some tricks and some hacks on Illumina sequencing, we can sequence the full length receptor sequence for the B-cell receptor, and that's all good. We can then make millions of reads, and we can generate hundreds of thousands of B-cell receptor sequences from like one individual, or maybe from even like hundreds, if that's what we want. But at the end of the day, we don't know what these receptors are binding, and so it's very difficult to say something about like the specific process between the antibody and the antigen. Mm-hmm. What we can say, or what we can do, is that we can take the sequences that we get from sequencing, and then we can start clustering them in such a way that we attempt to recapitulate the relationship between sequences that were also related biologically, because they all came from one recent common ancestor. We can try to recreate that by clustering them in a clever way.
Okay, so now I think we have enough background to formulate the problem, precisely the problem you're tackling, and I sort of alluded to it, but can you put it just concisely so that we have, we know what we are striving for?
Yeah. So there's a few concepts juggling around, and so there's affinity maturation, there's germinal centers, and then there's something that we got to introduce first, which is clonal families. And I think the whole problem here boils down to the fact that one germinal center can potentially host many naive cells. So back in the old days, we were thinking about germinal centers as like one. It was founded by just a single B cell. So that made everything very easy, because then all the cells that were in this germinal center, they were all from the same rearrangement event. They were all from the same common ancestor, like this single naive B cell. That turned out not to be true at all. It turns out that there's actually like a few 50, maybe 100, 150 naive B cells that are seeding these germinal centers. However, as it turns out, they usually actually resolve because there's heavy competition, and then one of those clones will win. And then we're close to the starting situation. But it's just to say that it's a complicated process. The way that we can make it more simple is that we can introduce a concept of a clonal family. And the clonal family here is just defined as all the sequences that are related to the same, so all the affinity matured sequences that have harbored lots of mutations, that are all descendants of a common ancestor, a common naive B cell. So with that concept introduced, what we attempted to make was an inference algorithm that could infer the substitution profile of such a clonal family.
Yeah, and substitution profile being...
Oh yeah, so that's a detail as well, that because we're interested in the selection process, which is in the germinal center. So we are interested in affinity maturation. That makes nucleotide substitutions irrelevant. At least if we make the assumption that all codons are equally good, then so none of these mutations they don't have any selective advantage. So, and that's the assumption that we're making. And therefore, all of these, when we're talking about substitutions, we are talking about substitutions in amino acid space.
Yeah, and so the profile itself is a set, like for every amino acid, right? It's a set of probabilities of, in this clonal family, how often do we see the substitution of this naive amino acid into another amino acid?
Yeah, correct. It's a position-wise substitution profile. So it's not just a substitution matrix where you can look up all the different, like going from one amino acid to the other, but it's a like per position propensity or whatever you want to call it. It's per position. So we're able to, because these receptors are fairly conserved, we are able to align them to the same alignment. So they can all be aligned in the same coordinate system. And therefore, it makes it quite easy to map substitutions back to a single position. And therefore, we can create these profiles for position number 13. There will be this percentage of alanine and this percentage of glycine, and et cetera, et cetera.
Yeah. And so before we go into the details of how you managed to do that, convince us that this is an interesting and important problem to work on. Like, and I guess I'm also interested in how you arrived at that problem. What motivated you in the first place?
Yeah. So the motivation was that we observed that when we do sequencing of like B-cell receptors via like a blood sample, then the clonal family size, so the number of sequences that we can map back to a single clonal family, that size distribution is following a power law. And what that basically means is that the majority of the clonal families will only have one or two observations. Actually, the majority will just have a single observation. So if you want to or if you happen to isolate a sequence that you are interested in because it binds something relevant, and it comes from a clonal family that only has a single sequence, well then it's not very easy to make informed choices about substitutions. And so from an engineering point of view, that makes it interesting.
Why is it important to know about this substitution? So presumably in the selection process, the sequence that won this competition, right, this is the best sequence. So why do you want to find out about all the other sequences that lost?
Because it's probably not. So I think that we can't necessarily say that it's a winner takes all game in these maturation processes. There is a lot of flexibility in these receptors. So some positions, it won't matter if you have threonine or serine on that position. It simply won't matter for the fitness. So again, my point of view is from the engineering point of view. I think I'm being biased because I worked in a company that made monoclonal antibodies for therapeutic use, and in that case, we did use B-cell receptor sequencing, and we did also face the problem that many of those clones just had a single sequence, and so if we were to make informed choices about like engineering that receptor, we didn't have much information from the sequencing other than that we could isolate that single sequence. So again, like it falls back on the engineering application, I think.
This is actually very interesting. Could you spell out that a bit? Like how do you use this in the engineering problem of like developing antibodies?
Yeah, so there's a massive interest in developing antibodies these days, and I mean, most people in the field of immunology and especially in the field of cancer, they know that there's a wave of PD-1 and PD-L1 and CTLA and all of these immune blockade antibodies that are being tested, and as it turns out, antibodies are just very convenient for making new therapeutics. You can design them to bind pretty much anything, so you can. If you have some biological process by which you want to make an agonist or an antagonist for, you might be able to do that with an antibody. So that sounds simple. Then you just need to make an antibody and needs to bind something. But as it then turns out, there is all these problems downstream in terms of like antibodies are prone to aggregating. So when you might be able to find an antibody that binds to the right antigen target, but if you need to produce it in in large amounts, and if you need to make it into a drug, it needs to go into a syringe, and it doesn't work out if it's if that solution is extremely viscous, so you can't like inject it. And the same with like something like stability, you need to have a stable protein. Same with expressibility, you need to have something that can be expressed, and et cetera, et cetera. So, I mean, there's just lots of complications. When you need to make a therapeutic antibody, you need to go through a ton of different quality control steps, and that's just very difficult. So the way that you can work your way around some of these classical problems about antibodies aggregating and antibodies being immunogenic, that's another big problem, is by introducing single substitutions. And you can do that sort of randomly and just try your luck and do all the assays to see if you got it right, or you can try to do it in an informed way. And I would say that this provides one informed way of introducing substitutions.
Yeah, so you can look at these profiles, at these frequencies at which the different substitutions occur, and presumably the B-cell receptor sequence that you get out of your sequencing will probably correspond to the most probable one, right? The most probable set of amino acids, but maybe you can trade that. Maybe you can settle for a bit less probable, and so maybe it's like a little bit worse, but it has these other independent good qualities that you're looking for.
Yeah, I mean, that's a good observation. Of course, you would expect that if you're drawing from a pool of sequences, you would expect to draw the most likely would be the highest abundant sequence in that set. But yeah, I mean, as it turns out, there are quite a few positions that they're just flexible by nature to some substitutions. But you might not know those a priori, so this just provides a more principled way of choosing such substitutions.
Cool. So now let's talk about the data that allows you to make these inferences. What sort of input data does your algorithm need?
I think we listed a couple of data sets here. 6 data sets, right?
Well, like in general, what kind of data is that? So I know you describe and you refer to the process or protocol called Rep-seq or repertoire sequencing. Could you maybe start with that? What is that and how does it work?
Yeah. So repertoire sequencing it's a process by which you take a sample. It could be blood. It could be you can dissect out a lymph node from a mouse, for instance, or it could be all sorts of immune compartments where you can find B cells. And then there's a process of isolating the B cell receptor sequences. And so you can go about this in 2 ways. You can either try to isolate it on DNA level or do the other thing that most people are doing, simply amplifying the transcripts, specifically with primers that are binding upstream and downstream of the variable region of this receptor. And then you'll get a PCR product, which is, about 450, maybe all the way up to 550 base pairs. So that doesn't fit into a single Illumina read, which is like 300 base pairs for my seq. But if you do paired end, you'll actually get a small overlap between the two, and then you can merge them on the middle, and then you'll have resolved your full receptor sequence with like a single paired end Illumina read. And you can like, to be fair, you can also do this on 454 platforms, and you might even be able to do it on Iron Torrent or something like that. But people are moving, or already have moved, to more or less purely just Illumina sequences, and then by doing that you can get like millions of reads for a single sample.
And so you mentioned that you can, for example, extract this lymph node out of a mouse. So will that be applicable to humans? How similar are the sequences between mice and humans?
Yeah. Of course, you can't just, you can't isolate the spleen of a human being, and you probably also can't just isolate a lymph node. I think you can probably isolate tonsils, but that's the only like lymph node or lymph compartment you can isolate from a human being unless they're dead. There are few studies where they use small like needles, and then they can pull out stuff from the lymph nodes with those needles. But most people, like 99% of these data sets, are just on whole blood.
Right, but can you use the data obtained from a mouse to enhance your resolution of your algorithm? Can you use that as one of inputs without sort of any bad side effects?
Yeah, that's well. So first of all, this is all human data sets, and and it's all from blood draws. So these are all B cells that are circulating in the blood, and so the immune compartments are all connected they're all in like there's the whole lymph system with lymph nodes and different compartments, but they all communicate with the peripheral blood. So I mean, it's a good question whether you can have, if you can extrapolate from one compartment to all the immune compartments, if you can extrapolate from the sequences you observe in the blood compartment to everything else. But I think there is a common agreement that there is a spillover between the different compartments, that if you have a clone which is residing in the gut somewhere, that you'll also be able to find at least few sequences in like circulating in the blood. But to answer the question about like if we use mouse sequences, no, we don't use mouse sequences because their B cell receptors are slightly different than human B cell receptors. Structurally they look similar, but the sequences are somewhat divergent. So we don't use that data to enhance anything in the human data sets.
Got it. Yeah. So another interesting choice that you mentioned is the choice between DNA sequencing and RNA sequencing. And so I was thinking about this myself. So I sort of see two advantages of RNA sequencing. So one advantage is that from what I understand, in a B cell, right? So there are two haplosets that each can encode for these B cell receptors, but only one of them is actually expressed, is active, and the other one is suppressed. So by sequencing RNA, you automatically get the right one, whereas if you sequence DNA, you would get both, and one of them is just not very good. And the other thing I thought about was that by sequencing RNA, you actually get more material, right? Because there's just one functional molecule of DNA, but it produces a lot of RNA molecules. And so when we sequence genomes, we can rely on bulk data because all of the cells have more or less the same genome. But once we zoom into this variable genes, we need to look at presumably the single cell resolution, right? So there's just too little material. But are there any advantages of DNA sequencing? And what are the pros and cons?
Yeah, I mean that's a couple of good observations. So yeah, just to recapitulate that of course there's two chromosomes and that encode for the B cell receptor sequence, and once we have this rearrangement event going on, there is something called allelic exclusion. So you only get expression from one of those loci. So yeah, you're right that on RNA level we will only observe, and that this is a slight. There's a bit of untrue in that, but in a general case, we'll only observe functional rearrangements, and so that's good. And we also get an amplification of our signal because there's maybe 1,000 or maybe even 10,000 RNA molecules in a single cell. But that's actually also a problem because we get an amplification of our signal, but B cells they come in different forms. So a naive B cell has a lower mRNA expression as, say, a plasma cell. So a plasma B cell, its basic job is to spit out antibodies in the fastest rate possible. So obviously it has also a very high transcription rate of the B cell receptor loci.
But that's a good thing, right? Because you're not very interested in the naive cells.
Well, sometimes we are, right? And sometimes we're also interested in the memory cells. So that's another thing. Mm-hmm. So memory cells could be related to, so that you could have a pool of cells and you could have a bunch of memory cells in there that are all related to a couple of plasma cells. So in the optimal case, you would really want to try to resolve that, and you would prefer to have sequences for both memory cells and plasma cells. But your problem in this case will be that if doing this on bulk messenger RNA, the plasma cells will make around 100-fold more mRNA than the memory cells, so they'll dilute out the memory cell signal, so to say. And also another thing is that you can't use transcript count as a good proxy for cells, for cell count, which is another problem.
That's a very good point. Yeah.
So I mean, but this is still the technique that most people are using. There are few groups. I'm not sure how many academic groups are doing it yet, but there's at least a company or two that are doing this with droplets, where they can definitely get single cell resolution, and they avoid the problem of not being able to resolve plasma cells versus memory cells.
So how do you work around this issue in your own work? Do you just discard the identical transcripts?
No, we just don't work with. We don't. We try to avoid having to use cell abundances, because we don't have a good way of estimating them unless we have single cell data. So, I mean, one way we can use this data is we don't want to throw out any data per se, but so if we observe multiple reads with the same sequence, then we just merge them.
But you have to rely on counts in order, because you're looking at the frequency profiles, right? So you need to know whether this amino acid has a frequency of 90%, either because it's a very frequent sort of variant, or because it's just present in the cell that is like very highly expresses its RNA, right? Yeah. So see, that's true. So
one way of looking at this is to say, well, if you have a very high fitness B cell receptor, then within our world we assume that all the synonymous mutations they don't confer any fitness advantage at all. So then we would expect to observe the same amino acid sequence, but with many synonymous mutation variants. And so that is the way that we can make like one amino acid being preferred compared to other amino acids, because like the highest fitness amino acid in our case would probably have a very high count, but it doesn't have a high count because it has many transcript counts per se, but because it has many different DNA sequence counts.
Okay, that's a good point. That's another reason why it could be a very high count, right? But this does not exclude the first reason, right? That's also a possibility, or is it not?
Say the first reason again.
So, like the first reason is just a high transcription rate, even if it's stochastic, right? It just could be very high by chance, and the other reason is, as you say, synonymous substitutions.
Yeah, but if there is a cell, so if there is a cell that has a very high transcript level, so say a plasma cell, then of course there is a chance that we will make an error in the sequencing, but otherwise it would be collapsed down to a single cell because it all has the same transcript.
Right, so that's what I meant when I say you just discard identical transcripts. So another way to say that you just merge them, right? But you discard all but one of that family of identical transcripts.
That's correct. And so one extra fact is that we're only using sequences here that have unique molecular identifiers. So that's our way of dealing with the fact that especially for RNA sequencing, you could be worried about the problem of having too many errors in your data, and that would inflate the number of sequences you observe with like observed synonymous mutations.
So when you decide to merge multiple transcripts, do you also look at the unique molecular identifiers, right? Okay, so
that's how we merge them actually.
Okay, so you will look at the different transcripts still as different sort of pieces of evidence for this frequency, right? They will all contribute.
Yeah. So the way that this is done is that there's a standard sequencing protocol with unique molecular identifiers, and you use that identifier to merge the transcripts, and then take a consensus sequence out of those merged sequences. And of course, if there is one sequence which is completely off, like by many substitutions, then it's either discarded or something else is done. But in general, what we observe is that there is a single unique molecular identifier where we can map back tens, maybe sometimes even hundreds of mRNA reads, and on average they will have one mismatch, one mismatch per read. And by taking the consensus, we think that we will have a pretty good estimate of the true underlying DNA sequence. And then actually we're making some cutoff saying that we don't want to include any sequences where we don't have enough evidence that to make our consensus sequence.
Okay. So this is how you get your training data for your model, right? You just take this repertoire sequencing data from, I guess, public sources, right? Did you just search on SRA for these data sets?
We get it from public sources, and I think all of these data sets are on SRA, but we have specifically contacted the authors of the articles where those data sets have been published to get the processed data, simply because there's so many ways of processing this B cell receptor data, and it all depends on which protocol you use for sequencing. So that I mean, we feel more confident that we're getting the pre-processed data from the authors themselves. Mm-hmm. And then, of course, we do all, on our side, we do all the validation, and sometimes we also apply extra filters, but in general, they know best about how to filter down the bad sequences.
And so, are you at all interested in the phenotypes for the sequencing data? So whether your sequences come from a healthy patient or a patient with disease 1 or a patient with disease 2?
In general, we haven't been so interested in it. We have been agnostic to phenotypes. We have data sets here that is before and after influenza vaccination. We have healthy individuals. We have individuals with autoimmune disease, myasthenia gravis, and we have here sequences from patients with West Nile virus and some different things in here. I think a common theme is that even though you have a disease like an autoimmune disease, or if you have been exposed to vaccination, then the repertoire is changing for sure, but there's still a lot of the same in the repertoire. So I think we can justify using data from various sources, unless it's something like an extreme case where you have complete immunodepletion or something like that.
Yeah, but also, couldn't that introduce bias in your data, like depending on which antigens you consider, depending on which antigens those antibodies are targeting, right? Presumably the profiles would look different, right?
Yeah, absolutely. But as I mentioned before, we're also agnostic to antigen binding, because we don't know what these are binding anyways. We just know they bind something, or they were elicited and matured towards some antigen. So we try to make summary statistics here. So we actually want to get data from as many different antigen sources as possible.
Right. That makes sense that you don't care about antigens, but it just could happen so that, for example, if you and I run the same algorithm, but on different data sets, so you happen to have access to patients with influenza, and I happen to have access to patients with cancer, right? And our profiles will be very different because those antigens generate very different antibodies. Even though, like, neither of us cares about the specific antigen.
Yeah. So, but that's why we have a model that is trained on so many different kinds of data, I guess. So, that is to attempt to be agnostic to antigens.
Right.
And so yeah. But I mean, you're right about that there could be some like bias there. But I would say the more data we include from the more sources as possible, then we're going to get a better and better summary statistics over.
But that also means that these frequencies, you're inferring the frequencies, right? And the frequencies themselves will be sensitive. Like, even if you get all the data you can and you have a very diverse set of data, still the proportions in which those data sets are present, right? They will influence the frequencies. So if you have condition 1 with one profile and condition 2 with the second profile, then if you mix them, then the profile presumably will be also a mix of those two profiles. And so depending on the proportion of the two cases in your data, in your total data set, right, your profile will be exactly dependent on those proportions of your input data.
It doesn't seem to be dependent very much. I mean, Amrit, you might be able to comment on this as well, but I think what we observe throughout, like, playing around with this data is that we have different ways of, like, of course, doing cross-validation, but we also have a complete, like, hold it out validation set. And we see, like, on a summary level, over, like, summarizing over all the clonal families, we see quite similar statistics. So I guess that sort of goes against the idea that there should be a very different repertoire between immunized versus non-immunized, or diseased versus non-diseased.
Okay, fair enough. So, and does that make sense at all to look at like healthy people? So presumably you would get what the memory B-cell receptors mainly.
Yeah, well, we already include healthy people, so.
Okay.
Yeah.
Right, and so that's your training set, right? And then also, so the goal of your algorithm to take in a new sequence, right? So because you're doing inference for a specific clonal family, so your algorithm takes as input a single sequence from that clonal family and tries to infer the profile for that specific clonal family, right?
Yeah. So that is our way of, like, we already have data sets where we observe large clonal families, and then we can just pick out one sequence, making like a subsample. We already know the true quote unquote substitution profile because we have this clonal family with many sequences.
Yeah.
And so we can make the inference on one sequence and then go back and look in the full clonal family to see if we can recapitulate that substitution profile.
So once you do this sequencing, right, you mentioned that you don't currently do a single cell protocol, right? So you have all this mass of all the sequences, although with the UMIs, and so then you have to reconstruct the clonal families, right? And also presumably you have to. I don't know. Do you do inference on the sort of the structure of the genes, so the V, D, and J regions? Mm-hmm. So what? Tell me what kind of pre-processing you do before applying. And so the spoiler is that ultimately you do this method that is called tensor regression, right? But before applying that tensor regression, you have to prepare your data. So walk me through the kind of preparation you're doing.
Yeah, you're right about that. This project is a lot about just processing the data so that it gets to the format that we can actually use for anything. And but I mean, I would say we're quite lucky because I think the flagship software of the Madsen group is this software that a postdoc in Eric's group has developed. So Duncan has developed a software called Partis that is, I think in broad strokes, it's a hidden marker model that can annotate and cluster sequences, B-cell receptor sequences, in a more like statistically principled way than most other people are currently doing. And so what we can do is that after all the processing, after all the merging and getting out consensus sequences, then we will feed those sequences into Partis, and Partis will start making like annotations. So it'll infer which genes were recombined, and it will infer something about this recombination process. And it'll use all that it'll use all that jointly to infer which sequences came from the same ancestor. And so that's the whole process of going from just unrelated data to sequences that are related with clonal family structures. And also by doing that, we'll get the naive sequence that we're actually also using in the inference as a feature.
Mm-hmm. Interesting. So, and how is that data represented? Is just a set of FASTA files, one per clonal family, or?
Now, so the input data to Partis is a FASTA file, obviously, and it's a FASTA file per individual. So of course you can't have a clonal family that shares sequences between individuals because for obvious reasons those sequences could not arise from the same common ancestor. Yeah. But and so therefore that's a logical breakpoint, and that has also been the breakpoint here. That whenever there is when we merge all the sequencing files, into a single file that represents one individual. And then all those sequences are getting run through Partis, and then Partis is spitting out a format, a CSV formatted file that contains identifiers for all the sequences that have been inferred to be in the same family, and then there is some information about the inference on the naive sequence and on the V gene and on the mutations, and there's just a lot of annotations embedded in that format.
Okay, got it. And then you also use something that's called AHO numbering scheme. So what is that, and how does that work?
Yeah, so that's going back to what I alluded to before about antibodies being quite conserved. So if you look at the three-dimensional structure, they're all very conserved. So antibodies are made. The variable region of the antibodies are made up of two regions. So there is the framework regions, and then there's the complementary determining regions, the CDR regions, and the CDRs are these loop regions that are fairly variable, and then you can mutate them, and they're flexible, and so those are usually also the ones that can bind different epitopes, so different antigens. And then there's the frameworks, and the frameworks are making up that structure which is common for all these immunoglobulin proteins. But long story short, because they're conserved structurally, you can make a numbering scheme so that you can number. You can take a sequence and then take a position and then map that back to from a one-dimensional sequence, map that back to a three-dimensional structure. And that's the idea here, is that whenever we refer to a number, when we refer to number 10, then that's an approximate position in three-dimensional space. And that's what's underlying this AHO numbering. I should say this is not it's not exclusively the only numbering scheme we could have used. It's just convenient in our application, but we could also have used another numbering scheme called Cabot or Clothia, or there's an IMDT numbering scheme. And so all these different numbering schemes were developed with that thing in mind, that you wanted to align a bunch of sequences, and the best way to align them is to prefer the position to the same place in a three-dimensional structure. And as it turns out, this AHO numbering is just the most convenient one because it's in a format where pretty much all sequences, unless they look very odd, will have the same vector of numbers. And we like to work with a fixed length vector when we do statistics.
Yeah, so there are in total 149 of those positions, right?
Positions, yeah.
And so
many of them are gaps, actually, so they'll be regarded a little bit special. But it's just to say that we input a sequence, and we always we consistently get out a vector of 149 positions, and that's going to be very important if we want to make some principal statistics on it.
Right. So this vector is a vector of amino acids, right? Each for one position.
Yeah.
Yeah. But some of them could be missing.
Yeah, many of them could be missing because, as should be clear now, antibodies are extremely variable. So some antibodies have, in those loop regions that are very variable, you can have some loops that are longer than others. So there's some space to put in longer loop regions and shorter loop regions, and the way to do that in such a numbering scheme will be to have some way of introducing gaps, and there's a principal way of introducing those gaps.
Right, and these positions are inferred based on sequence alignment, or is there some kind of 3D protein modeling going on?
Yeah, it's going back to this three-dimensional mapping back from a sequence to a three-dimensional structure. So the way the Dhar numbering scheme and like the general principle behind those numbering schemes for antibodies is that that you can input any sequence, and then when you refer to position 10, then that is approximately the same position in the three-dimensional structure for all different sequences. And the way that they can do this is that they can make this numbering scheme, and then they can optimize on like they can minimize the observed difference in three-dimensional distance, and that's how they do that.
Got it. Does that take then like very long time because for each single sequence you have to infer its 3D structure?
No, because there's an approximation for this, and that's a hidden macro model. So so when you have like, say, what is it? We are now around like 500, maybe 600 antibody sequences where we have the solved protein structure. Then you can easily do the numbering for those because you know how to map back a position in the sequence to a position in the three-dimensional structure. And so then once you have all those sequences aligned, then those 600 sequences aligned in a multiple sequence alignment, you can run a hidden macro, or you can train a hidden macro model on those, and then you'll get something that you can use for annotation afterwards.
Okay. And so
that's actually extremely fast.
Mm-hmm. So presumably once you have this annotation with echo, you know where each region, so the V, D, and J regions, where they are, right? But do you use that information at all, or do you just rely on the numbers?
Yeah, we rely on the numbers, but we also know where the VJJ positions are. That's output from the first annotation step, the parties annotation step.
Oh, right. Okay. Right. Cool. And so this is then ready to be fed into the tensor regression framework, right? So what is tensor regression? That's
for Amrit to...
Yeah, sure. Great. I mean, tensor regression, in general, it's kind of it's a regression when you have kind of multivariate kind of like parameter spaces. And you don't necessarily have a predictor matrix, but you have let's say a predictor cube. Common examples in like fMRI data, when you have these voxels, three-dimensional cubes of voxels kind of thing, and you want to like use that as a feature. And like tensor regression is like a way of kind of incorporating that kind of information in a principled manner. So, the common way of like doing analysis or whatever is just saying, okay, I have an image, let's say, and I just want, I just basically take away all the, I just basically collapse everything into a row and make that my feature space, right? And that you can do that, and that's great. But the tensor regression kind of does it in a principled manner where it doesn't actually, in the most simplistic manner, it'll be, let's collapse everything to a row. But for more, it's within a general framework, it won't actually decompose the cube into a row, but it'll just it'll preserve the spatial properties of the predictor data. And in our case, we kind of have that. We don't necessarily need to take advantage of all the good features of tensor regression, but certainly our data is multidimensional, in the sense that, we have, one dimension being all the clonal families, right? The other dimension being, so we have imagine we have a matrix, right? After all this AHO numbering and, processing of the data, like our rows are, let's say, the number of clonal families, each row being a clonal family. And let's say the columns, you could presume that, the columns are each AHO position, 1 to 149, right? But of course, in each of these cells, we need to have an entire vector, right? A vector for the amino acid probabilities. So right there, it's like, oh, well, this isn't really a matrix. This is actually a cube. So we can't necessarily, do regression in the kind of classical sense. It's a little harder. But, given the scientific, like, for instance, we wanted to make the model a site-wise model. So we had very specific constraints where we said, we want to do kind of our regression model, we want it to be kind of a weighted average of information across sites.
Can you explain what a site-wide site-wise model is?
Yeah. So basically, we have all these information sources. So, Kristian talked a lot about the processing and kind of what we ended up, getting. The data that we have we use is kind of, we have lots of different profiles. So basically we have, so site-wise model is basically, you have a particular site in an AHO alignment, and you're asking the question, what can we make a weighted average combining different data sources at an AHO position and try and predict, the AHO position, amino acid frequencies in a particular, input sequence. So, and you want to do that site by site. So we get these kind of like site-wise amino acid, preferences.
Got it. So, when you say tensor, you essentially mean just a multi-dimensional array, something like that?
Yeah, exactly. Yeah. Tensor and array are we're not in the physics sense of tensors, but definitely, it's basically an array.
Yeah, but also, like, I'm curious, is there at all connection to this mathematical tensors, which are, like, sure, they're multidimensional array, but there is more structure to them, right? Because you can sort of change the coordinates and stuff like that. Is there any connection, or these are just multidimensional arrays?
No, I mean, these are just basically multidimensional arrays. I think it was important to frame it this way because we couldn't necessarily certain the arrays needed to have the dimensions. Otherwise, it would just be kind of a flat array. And it didn't make sense to be flat because you couldn't compare them each element equally, depending on what dimension they were. And at least in our case, we didn't necessarily need to exploit a lot of those properties, like special properties of tensors. Because in our case, we were doing something very specific, which was at each site, compute a weighted average and do it for all sites. So basically, like for a particular dimension in our tensor, compute a weighted average and do this across all elements of that dimension. So it didn't really require much more than just keeping it in the array, all the information in the array.
So at each site, you take a weighted average, and so you are looking for the weights, right? So your regression results in the set of weights, and the weights themselves are site-specific. Is that correct, or are they universal?
Yes, So kind of how the weight matrix works is, you have a set of weights for a set of unique weights for each site, and at each site, depending on how many information sources you use, clonal, substitution profiles you use, that determines how many per site weights there are. So for instance, if I'm using, let's say, four sources of information, substitution profile sources, and that means that at each site there'll be four kind of regression weights.
Mm-hmm.
And so each information source gets its own weight, and then the input sequence gets is like 1 minus its weight. So we want all the weights to sum to 1. Yeah.
Right. So this sounds like essentially a linear regression, right? If you sort of forget about the tensor stuff, is this just a linear regression and with the regularization?
Yeah, you could see it that way. I like thinking of it as a convex optimization problem with maybe more constraints than linear regression. Certainly there's the regularization part, which looks familiar to people who've seen machine learning stuff, and, Lasso and Lars and things of that nature. But, in our case, we have very specific constraints that all the weights need to be between 0 and 1, and the sum needs to be constrained to be equal to 1.
Right.
So, it's a little hard. I mean, yes, it's very similar to linear regression, because it is a weighted, it is a linear combination of parameter times data, and then basically summing parameter times data over and over, right? And that's how you get the site-wise model. But then, then you have a little more constraints because the weights have kind of unique interpretations by having to sum to 1.
Okay, so talk about these different data sources, because on the one hand, we may have different data sets. As you said, you contacted different people, right? Got the results of different experiments from them. So presumably these are different data sources, but also in the paper, you sort of do a classification. So you have a V gene cluster, V subgroup cluster. So what are those things, and how they interact with the different data sets?
Yeah. So as Kristian was talking about, we had 5 or 6 different data sources. 6 actually. Yeah. And the amazing part about it, you and Kristian were talking about whether it was good or bad to have the data be non-specific or specific for a particular antigen or anything. And I actually think it's pretty remarkable that, our data set's pretty broad. As Kristian said, it's got a lot of different, antibody information from a lot of different viruses and autoimmune diseases, whatever, right? And what's great is that we use this data, this kind of publicly, this public repertoire data for both training and testing, right? So we're not touching anything else. We're purely just touching this kind of database of information, this public database of information we have. And somehow we're able to, get kind of predictions that are better than what have happened before, which is kind of crazy. And so Kristian said we have this public data, and how we got this data set is, in particular, we wanted, for inference, right? The goal of our project was, if I have a B cell receptor sequence, and, we don't necessarily know its clonal family, can I predict its preferences, right? Its amino acid preferences at each site. So kind of how we recapitulated that is we tried to take the largest clonal families in this public repertoire data set. And the reason we took the largest was because we wanted to have, the greatest amount of information. So I believe we tried to pick the largest clonal families that had more than 100 sequences in them, mainly because we wanted to treat these as kind of a ground truth data set for inference. So what we did was, for each of these clonal families, we would subsample a single sequence from them. So the full kind of, so in these clonal families, they have more than 100 sequences, maybe 1,000 sequences, but definitely more than 100. And so that kind of serves as ground truth. We know the answer. We know what the true substitution profile looks like. We just observe the frequencies from affinity maturation and from sequencing, and that should reflect the actual amino acid preferences. And then we subsample a single sequence, and then we ask the question, okay, from the subsampled sequence, can we somehow recapitulate this entire clonal family amino acid composition? And so we do this process until we got 500 clonal families, and we use that as our inference data set. And then all the other kind of clonal families that were not chosen for inference were used as our kind of public, what I'll also call query data set. It was a data set that we used basically to help with the inference. And so getting back to, I know it was a long-winded answer, but getting back to kind of what our data sources are, we had 4 main sources of information, so of course you have the input sequence, which is kind of what you feed into the kind of the model, right? And then, the 4 main sources of information we had were, we had the naive amino acid sequence, which is obtained by Pardis, as Kristian mentioned before. But
from this input sequence, right?
Yes, exactly. Yes. Pardis is rerun on this input sequence later on. Yeah. So in particular, the Pardis has no other information about the clonal family beyond the input sequence. So we're not cheating in any way.
But it is also it has a trained model inside it, right? Because I could imagine if you give to Pardis a big clonal family, it could sort of look at all the sequences and guess what the naive sequence was, right? But if you give it just a single sequence, it has to know, like in general, from some pre-trained model, what the possibilities are.
I agree with you. And this actually came up, as we were writing the paper. I was thinking to myself, like, how is the inference done? And it turns out that Duncan has, a kind of pre-specified parameter set that in practice works really well and has nothing to do with our particular task at hand. Which still, so in some sense, we're not actually using any kind of built-in information from our data sets. We're just using kind of pre-cached parameters that Duncan has found to be empirically useful. Which in some sense is kind of also remarkable because, it's still giving us good naive sequence inferences, despite the fact that we're not actually training the HMM, in Pardis itself. We're just using pre-cached parameters from a different data set.
And so if this model is really good, then you could take a big clonal families, right, and feed to Pardis, like each sequence one by one, and in theory, it should produce a single naive sequence for all of... Although, on the other hand, of course, it was Pardis itself who clustered, right, these...
Yes.
The sequences into clonal families. Mm-hmm. So, but if you could somehow sequence a single clonal family, then you could verify that Pardis gives a single naive sequence for the whole family.
Yeah, so Pardis does a lot of things. So I think using the word Pardis doesn't really do it justice because it's like Pardis, it does annotation, so it does do the naive sequence inference, and then it evolved to do clustering as well. So clustering was kind of the next step. And then it evolved to do even like germline gene inference and, so kind of the naive sequence inference was like the first thing. You could feed it in a single sequence or multiple sequences and it will give you some sort of, naive sequence. But, the first step was to do the clustering, which was like, just how we got our clonal families. But then once we subsample a sequence, from these clonal families, it doesn't, the subsampled sequence doesn't really know anything about its members. And then we just, run it through Pardis and we say, we specify when we run Pardis, like we want to only annotate the sequence with a naive sequence, but no more clustering. Mm-hmm. Like that functionality we did already.
Okay. So I guess the two data sources are the naive sequence and input sequence, right? So what about the others?
Yeah. So kind of the public, I think the two input sources that are related to the public repertoire data, which I think is the most, right? Is what I think we put a lot of emphasis on is the kind of what we call the V gene kind of substitution profiles and the V subgroup substitution profiles. So kind of what we do is, Pardis is, clearly we're very, we use a lot of Pardis information here, but Pardis also provides, inferred V, D, and J genes, as output as well in its annotation. And we use that information to basically help us in our inference. So kind of what we do is we look in our we look in our input sequences and we ask Pardis, we ask Pardis, okay, can you tell me my V, D, and J gene, right? And so then Pardis in particular, maybe Kristian can talk about, we use a lot of V gene information, V subgroup and V gene, which is, in case you're not aware of, antibodies, the V gene spans most of the antibody sequence. I would say that, it's a fairly conserved sequence, but, most of the diversity of the antibody is like within the small CDR3 region. that where the V, D, and J kind of have a little bit of influence all in. But kind of what we do is we look at the public repertoire data and we say, okay, why don't we try and create profiles in the public repertoire data that we've excluded from inference testing, and why don't we just group profiles based on like their V gene and V subgroup label, and in particular, what is a V subgroup and V gene? Well, Kristian, correct me if I'm wrong here. It's a particular labeling scheme from IMGT that kind of determines kind of the relationships between kind of V genes based on certain schemes that they've developed. And a V subgroup is a very generic group. It's kind of a higher level group that contains lots of it's basically the first division of V genes. You first look at the different subgroups. It could be V1, V2, V3, and that's kind of the first denomination of kind of V genes. And then you kind of go more, in a more fine-grained nature, you divide into V genes, which is kind of it's a weird term to call it, V gene, but, it's like a V gene would be V1-2 or V1-3. So that's a more fine-grained label for the V gene. And so, what we do in our methods is that we actually cluster the public repertoire data based on these labels, these inferred labels, to kind of act as a kind of different levels of cluster information, right? So like the V subgroup cluster information provides us a very high level of profile of VDJ profile information, whereas the V gene information might give us very, more niche, more kind of detailed cluster, like profile information specific to each V gene. And we thought, both of them, certain kind of serve different purposes, one for high level, one for low level. And we found they're both very useful in the inference.
So if I understand correctly, in both cases with V gene and V subgroup gene, you take the all the public data, right? And it is already grouped into clonal families, but then you cluster those clonal families themselves. You join them, right? Based on the identity of a part of the sequence, whether that's yes. V gene and V subgroup both refer to like specific parts of sequences. Is that correct?
Yeah, I mean, so there's a particular part of the sequence that's the V gene, and we can kind of categorize these V genes based on certain particular labeling schemes. And the particular labeling scheme we use is from IMGT, and they have a kind of a scheme that talks about subgroups and genes. And so what we do is, like you said, we categorize these clonal families by the particular labeling schemes, and then we aggregate the profiles over those schemes, over those labels.
Right. Just to make sure. So when you say categorize, yes, based on the scheme, so you are talking about categorizing not the genes, like not the V1, V2, V3, but like specific alleles of V1, V2, V3, right? It's a categorization scheme for the alleles or for the sequences of the gene.
I do not know. Kristian, do you know the answer to that?
Yeah, maybe I should jump in. You're correct. It's on like the alleles. I think it's the basic. It's the basic notion about that the immunoglobulin loci are extremely variable. So a human will have somewhere between, I think, 40 and 30 genes, and so those V genes will be spatially separated out. But if you take gene number 1, like going down the chromosome, then that can have different alleles. So my V gene 1 allele might be different than your V gene 1 allele just by a single substitution. And so another way, and then going even, like categorizing them even, more coarse-grained, you could make these subgroups. And so that's just a definition so that you can make a more like coarse-grained grouping of the genes. So going back to your question or your comment, you're correct that the V gene cluster is when we refer to a V gene cluster, it's V gene 1 and it's all the alleles of V gene 1. And when we refer to V subgroup cluster, it's the subgroup and all the genes contained in that subgroup and all the allele contained in all those genes. So it's like a hierarchical classification, and everything which is under that hierarchy is defined in this labeling.
So if I understand correctly, V clustering means we have this V1, V2, V3, which are like different loci on the chromosome, right? Different parts of the gene. And in each clonal family, only one of those gets used, right?
Yeah, by definition, you can only, like, a clonal family is defined by a single naive sequence. So that naive sequence can, by definition, only have one V gene allele.
Right, and so you cluster together all the clonal families that use V1, right? That's your one cluster. Then you cluster all that use V2. That's your second cluster, something like that. Yeah,
so going back to the definition of subgroups and genes, then let's say V gene 1, that's the subgroup classification, and then V gene 1-1 is the gene classification. So then on the subgroup level, we cluster all the clonal families that are classified as V gene 1. And on the gene level clustering, we cluster all the clonal families that are using V gene 1-1. And so all the alleles that are in V gene 1-1.
Okay, got it. But despite the fact that you cluster based on the, which V gene gets used, you still, like, within the cluster, you still infer the full profile, not just the profile for the V gene, right, but the profile for the whole B-cell receptor sequence.
Yes, correct. So, and we can actually, it's very clear to see then, because that's a brilliant point, and the way that I think we can see that this is true is by looking at the contribution, because as you mentioned, it is like a regression model. So our weighting schemes here, like any old regression model, then the weightings, the alphas here, they represent how much of a given input feature is getting used. And the V gene is covering from framework 1 to the end of framework 3. And that is exactly also where we have the most contribution of the V gene and the V subgroup clusters. Beyond framework 3, there's pretty much no contribution from those two, which makes sense because the V gene is not defined in those regions.
Interesting, yeah. And so we've covered so far 4 sources, right, that you use. So the naive sequence, the input sequence, the V gene cluster, V subgroup cluster, right? But so there was the 5th one, which is the neutral substitution profile.
Yeah. So that comes from the fact that we know quite a lot about this substitution process. As I was explaining in the beginning, it's called somatic hypermutation, and it's a fairly complicated process. But long story short, it's a process which is it's a mutation process which is enhanced by an enzyme called AID, and that enzyme goes in and then it deaminates cytosines, and it does that in a motif, like in a biased way. So presumably it binds better and deaminates better around certain sequence contexts than other contexts. And also the repair machinery that comes and repairs it afterwards is maybe also biased. In it's not fully known how these biases are working out, but we can use summary statistics to infer what the substitution and what the mutation and substitution bias is on a neutral level. So we can do that with passenger alleles, and there's been much work going into that. And so the idea behind using a neutral substitution profile is simply to capture some of that information. And the way we can do that is simply by looking up in this motif-based mutation model, which is already that is inferred for humans already. And then we can simply just simulate mutations draw mutations and introduce them into the naive sequence, and then simulate a neutral substitution profile as we would expect it to look when there's no selection.
Right. And so the neutral substitution profile is based on the input sequence, right?
Yeah, it's
not general.
It's based on the inferred naive amino acid sequence.
Mm-hmm. Okay, got it. So these 5 information sources, right? But you don't make a distinction between different data sources, right? So you may have data from different experiments, different Rep-seq experiments, but they would share the weights as long as they fall into the same clusters.
Yeah, there's no discrimination between data sets. That's true. I think we pretty much rely on the fact that we make an external validation in the end on the best of all the data sets that we have, which actually also turns out to be a single cell data set. So it's also adding to the question before about that we can't count transcripts as single cells, but we can actually do that with the validation data set that we're using.
Yeah, and so then you have this model which combines data sources with this coefficients, and the coefficients vary among sites and among information sources, right? And then you apply regularization. So can you talk about the regularization you apply?
Yeah, Amrit.
Yeah, I'll jump in here. Obviously we could take our weighted average model at each site, compare this kind of weighted average predicted amino acid substitution profile, compare that to the real one from the real clonal family, right? And then maybe compute some error and then minimize that error, right? That's kind of the classical way of doing, statistical inference, right? And you could do that, right? No one stopped us from doing that. But I think there's a lot of noise, right? You could imagine little perturbations in the data set might change weights here and there. And so you don't really get a very interpretable signal, from fitting just a model with, tons of parameters, right? So in our case, you have 149 positions, and let's say we used, 4 unique external data sources. So that's 149 times 4, which is, I believe, is 596 unique parameters that we're estimating, which is a lot. So when you're doing the inference, it's we want to be able to understand what these weights mean, we want to understand, how do these weights, which sources provide the most information? How do they relate from site to site? And it's a bit difficult, whenever because it's so specific to one data set, and you want to be able to generalize your weights to new data sets. Regularization is a great way to do that, right? Instead of having a very jaggedy likelihood surface, regularization kind of adds some smoothness in this case, it's not a likelihood surface, but in this case, it's just kind of an error surface, right? And the smoothness acts as a kind of as a way to make sure that you're not overfitting to a particular, specific nature, specific kind of thing of your data set. right? Like presumably, you don't want to condition on the fact that your data set is the only data set you ever see, right? You want to be able to look at other data sets and have them be, variable nature of other data sets, have your model react to that, right? So, regularization is a common way of doing that. And so in particular, the regularization that we do use in our kind of SPURF model has been discussed in kind of machine learning quite a bit, and it has to do with lasso penalties. And kind of what a lasso penalty is well, basically it's kind of interesting because it will shoot kind of parameter estimates or parameter weights to zero if they're uninformative, which is kind of useful for interpretation because immediately that that something is not important if it's at zero, right? Versus other maybe regularization schemes which may kind of slowly converge to zero, and eventually will reach zero, but not like kind of hitting zero at a much much faster. And so that's kind of what we do in our model. And so, yeah, does that, do you want me to talk specifically about kind of the different, the two lasso penalties we use?
Yeah, so I'm trying to understand, you say there are a lot of parameters, and sure there are because there are many sites, right? But at each specific site, there are just 4 or 5 parameters. So, mm-hmm. and they sum up to 1. Mm-hmm. So you can't reduce that too much, right? Maybe you can drive maybe, I don't know, 1 or 2 to 0.
Well, yeah. So, I mean, we put penalties in all the parameters, but we all, we of course have to enforce the fact that they all sum to 1. But, in the case of, there were a lot of positions that I think in our plots that where, a lot of the, coefficients were at 0. And kind of, so we put a kind of a regular lasso coefficient for all 4 at each site, for all 4 of the information sources, to allow them to adjust to the data as need be.
So the sparse model, the result of your lasso regularization, would look like at each site out of 4 parameters, mm-hmm. 1 or 2 will be mm-hmm. 0. Is that?
Yeah, that's the hope, right? Like if truly that 1 or 2 parameter, only 2, let's say 2 information sources are relevant and the other 2 are just non-informative, then the hope is that the lasso will push those uninformative coefficients to 0.
But also the hope is that there is no information source that is irrelevant everywhere. So sort of at different sites, the different set of sources are relevant.
Yes. Of course, right. But you would hope that there's no information source that's uniformly uninformative across, all the AHO positions, which luckily was not the case. Because for you could imagine that, as I said before, the V gene spans, 70% of the antibody sequence. I don't know. Kristian can correct me. Sounds reasonable, right? And so you could imagine the V gene and V subgroup profiles being really informative for, 70% of the AHO positions because it spans that V gene, right? But right after that V gene, it not being important. And then, the other 2 kind of information sources, the naive sequence and the neutral profiles, those are kind of more specific to the entire, sequence, right? So you can imagine those sequences being more informative at the spots where the V gene kind of ceases to exist. So kind of intuitively, we did expect that there were going to be different areas where we thought that the information sources had higher weights, and we didn't expect, that any of them were going to be uniformly, 0, have weights of 0 kind of across the AHO alignment.
Yeah, that makes sense. So, and did you choose the sort of the weight of the regularization by cross-validation?
Yeah, I mean, it was a bit of an involved process. So, I mean, I kind of left out kind of, if you want me to, so we can go into detail, but kind of what we did. So, I've been very specific about, we used 4 information sources, right? But in reality, we had a couple more. We also used k-means clustering, and based on a couple. So, instead of using those V subgroup and V gene based clusters, we segmented those, these substitution profiles based on V subgroup and V gene labels. We also used k-means clustering to segment these public repertoire substitution profiles based on the naive sequence and based on just the profiles themselves. And those are 2 other sources that we wanted to investigate as we came to we wanted to figure out what we wanted to put in the model. And so what we did to kind of test what features we wanted to add into the model was we came up with a forward stepwise selection procedure. And the reason for it was because we certainly didn't want to do all subsets regression where we tested every possible combination of data sources in the model, as it would have wasted a little too much time. And, but we also wanted kind of an acceptable and kind of like something that would provide a good accuracy and also good efficiency trade-offs. So we came up with this stepwise selection procedure, which kind of worked as follows. Like, we would try a model, we would fit a model. So we would start with the null model where there's nothing in it. The model, it's kind of a set of 0, right? An empty set. And then we would throw in an information source. We would check all the first level, we would throw in the first profile in, and we would check over all the external profiles and measure the performance of all of them. And the best external profile, let's say, was the naive sequence profile. That would be, then we would say, okay, great. Continue on with that profile. So then we'd go to the next step, and we would fit a model where we had the naive sequence profile, and then we would look over all the possible profiles that were left in the set, excluding the naive sequence profile. And we would try those kind of models. These are second level models. And we kept doing that until you can keep doing that until you reach kind of a maximum threshold that you feel comfortable with. We chose 5. And we ended up just doing really good with 4. And that's how we got to, the 4 kind of the 4 external information sources that I was describing before. And in particular, how we're estimating error is we are doing 5-fold cross-validation within each step of the stepwise selection process.
Can you also just mention how we fit the regularization parameters? Like the lambdas.
Oh yes, but importantly, this is first, right?
Yeah.
It's a bit complicated. We first have to determine what, like, certainly we can't regularize until we know what discrete. So you could imagine like our kind of model selection process as optimizing over discrete parameters, where the discrete parameters is whether to include these information sources in, and the continuous parameters associated with regularization, right? And these are all tuning parameters that we need to like figure out, right? So kind of the first step was, we wanted to do forward stepwise selection to get to deal with the discrete parameters, which was tuning, do I include a information source or not? And once we determined the best kind of choice of information sources, we then ran. We then did kind of the regularized model fitting, which was, okay, now conditioning on those information sources, now let's do kind of cross-validation to determine the tuning, the tuning parameters from the regularized model. And then we did that, and we did cross-validation, we got, estimates, and then once we did that, we fit we fit the penalized model using the optimal tuning parameters, and then we got our parameter estimates of our alpha weights. And then once you have the alpha weights, you can then predict on basically any data set you want. So it's a bit of a kind of it's a very complicated and like involved process because of all the different kind of tuning parameters that exist in our model.
Right. So if I download the SPURF software, will it already contain this fitted alpha values?
Yes, it will. Yeah. It's
Cool.
Yeah. I mean, yeah. It's. I believe I don't know if we actually. I know the prediction. The tool will definitely. You use the alpha values that we provided. I don't know necessarily if it's actually in there. It probably is in the repository.
It is. Yeah, it sure is.
Yeah.
And
I must have uploaded it a while ago.
No, it's there, and it's it's also in the Docker image, so you can easily try it out just by downloading the Docker image and try it on a your favorite sequence.
Right, and the cool thing is, if I understand correctly, right, although you have to analyze this presumably huge amount of data because you have all this sequencing data, but in order to run the model, you just need this 4 profiles, right? So they're tiny data.
Yeah, well, you don't even. Yeah, you only need to have the parameters, which are not very. I mean, that's just a matrix, so that's nothing. And then you need a bit of. So you need the profiles, the input profiles, and it's only 2 of them that are based on the public data set.
Yeah, so all the original public data sets are huge, but the profiles are tiny, right?
Yeah, it's condensed into a megabyte or something like that.
And in particular, like our SPURF tool does that kind of profile generation while it does the prediction.
What do you mean by that?
I mean, like in the sense that like it'll determine. We have these clusters kind of cached, sitting there, and like when you input a sequence, it determines. It first runs Pardis, gets an I sequence, and then it determines the V gene, D gene, J gene, and then you use those the V gene to determine what subgroup cluster belongs to, what V gene cluster belongs to. It simulates a neutral substitution profile.
Right.
That's what I mean. It kind of the SPURF tool kind of does all that behind the scenes.
Cool. And can you talk just very briefly about how you actually implemented this? So this is an optimization problem, right? Hopefully a convex one.
Yeah.
But how do you actually go about do you code everything by hand, or do you use some kind of framework?
Yeah, I mean, so So in particular, I think I don't know. It's me and Kristian. Yeah, I think the people in the Madsen group really love Python, and I think a lot of the data processing tools have been written in Python. So a lot of the data processing when it came to preparing data and doing the AHO annotation was all in Python. And in terms of the model fitting itself, I'm an R guy, so when I was doing a lot of this model fitting stuff, I used a linear algebra library in C++, and then which was good for because it used some template meta programming. So I didn't have to worry about kind of like long expressions being kind of wasted at runtime. And so I would use this kind of like templated C++ code and just write out whatever kind of like objective functions that I needed to use, and then we use kind of a generic optimizer to fit these functions. I mean, so as you were alluding to, yes, our problem is convex because the lasso penalty is a convex function. With respect to the parameter, it's optimizing, right?
Yeah, lasso sure, but what about the error itself, the objective?
Oh yeah, I mean, so on the one, so we didn't actually really talk about that, but we actually used 2 different error objectives. The first error objective was the kind of the standard you would use in linear regression, right? It's the L2 error. We would just take kind of an L2 norm, a squared L2 norm of these profiles, and so as you can imagine, that is convex because it's a linear combination of of these terms, and it's an L2 norm, right?
But also you have this constraint that it sums up to 1, but I guess that's also fine because that's linear.
Yes, exactly. So in particular, these all need to be satisfy inequality constraints. So yeah, so the equality constraint fits, and the being less than or equal to 1 and greater than or equal to 0 also fits in kind of a convex optimization framework.
Yeah.
And so in that case, one may ask, especially in the statistical community, did you, could you come up with a kind of a specific solver just for this problem? And I think in our case, it's like, it just didn't really matter. Of course, the first level, to try is always, can you use something generic and does it work good enough? Right? Like, why waste time trying to engineer like a very unique optimization solver if generic solvers will work? And it turns out, we used a generic optimization solver even for a convex optimization problem, and it worked really well, which was great. We were concerned about, getting good results, not whether our optimization procedure was unique or something, because that was the real,
sure.
There's a lot of people who focus on that stuff, and that's great. But, we were really concerned about, getting good predictions, and whether if we could converge. We knew that at least for the L2 error, that we were going to have a unique solution because of the convexity. So it didn't really matter. We tried different restarts, and if it was converging to the same solution, we're pretty convinced that, that the generic optimizer would be just fine.
Yeah. And so that's presumably based on some kind of gradient method, right? And,
yes, exactly.
Do you use the approximate gradient based on finite differences, or because some frameworks can do this sort of automatic differentiation where they can?
No, yeah, we're doing none of that. We're doing numerical gradients. In particular, we were using the very classical LBFGS. So it's BFGS is that kind of it tried to approximate Newton's method by kind of doing Newton steps, but instead of using the actual Hessian, it uses kind of an approximate Hessian based off of like first order differences. And it, it works, you could actually supply a derivative function, but if you don't supply a derivative function, it'll just compute first order differences numerically. Yeah. And then it'll use those numerical first order differences to numerically estimate Hessians. And so that's kind of how it works. It's kind of a Newton's method, but it's kind of it's in the family of quasi-Newton optimization methods. And what's great about it, at least from our perspective, was that it also incorporates bound constraints, which we needed because the alpha parameters had to be constrained between 0 and 1.
Oh, that makes sense. Yeah. And so you mentioned the two objective functions. So one, we discussed the L2 metric. Can you briefly talk about the other one?
Yeah. So the other, as a statistician, I was pretty just content with L2 because, that's what I'm used to. But I think as kind of from a scientific point of view, I think there was some interest in understanding like, well, okay, so L2 is measuring like how good do these frequencies match up, right? Do these frequencies match up in an L2 norm sense? But like if some of the, like maybe, so scientists would only ask like for people who might use this tool, they might really be asking like, okay, so maybe what about the amino acid bases that, are greater than 20% or like, are observed more than 20%. Those are probably the only bases that I'll actually be looking at because if I want to mutate like when I'm doing engineering, I may only want to mutate, to amino acids that are greater than 20%, right? So, the question is, can I predict highly frequent amino acids, versus just kind of like the kind of the less frequent amino acids that they may not care about. So in this case, instead of comparing the entire kind of profile vector, we compared sets. So basically we had a set. Let's say I was interested in the amino acid frequencies that were above 20%, right? And I would, I would compute a predicted set and I would look at the actual set, right? So I'd have these sets that were consisted of the amino acids that were observed or predicted more than 20% of the time. And then I would compare them for their similarity. I would say, well, how close are the sets? Because, a scientist really wants to know, like, for these, I want to know what bases I can mutate to, and are you good at determining those bases that are highly frequent? And, there's a similarity metric that determines similarity based on sets called the Jaccard similarity metric. And just very simply, it's very simple metric. It's defined as the ratio of the intersection of two sets over the union of two sets. So it's kind of like it's the ratio of the intersection to the union. So it's a very intuitive metric. So if the two sets are the same, there's 100% similarity, which is great. If the intersection there's no intersection, that means that the similarity is 0, which also makes sense. So it's a kind of intuitive metric, and there is mathematical precedence for using this metric when you want to compare sets. So we found that to be kind of an interesting, kind of a good use case for the kind of a different metric to optimize over that may be of interest to scientists.
Yeah, and did you find that the inferred parameters differ too much between those two metrics?
I mean, I think the inferred parameters they do differ because of kind of what they're optimizing over. So, you could imagine like tweaking, some of the weights, which imagine the weights are like knobs, right? And you're tweaking these knobs, and like you could imagine, okay, I changed these weights a little bit, but if the sets, it's very easy to change these weights and have the sets remain the same, right? Because the weights are continuous parameters, but these sets are based off of like these thresholds, like am I greater than 20%, right? So it's very easy to get into a situation where, there's kind of a flatness where you're changing parameters in the space of parameters, right? But you're kind of still achieving the same sets, and so thus you're still achieving the same error.
Right, and that's presumably when the regularization kicks in.
Yeah, I mean,
to resolve those ties.
Yeah, but I think like even more like the regularization does help, but it's almost as if it's too like because of this discrete nature of the Jaccard, it's almost too smooth. I think the fact is like you can't, you move around in parameter space and it's so flat because little changes. I think LBFGS doesn't do large scale changes every iteration. It does, kind of gradual. So what's happening is that, we found that because LBFGS is gradient based, we were finding that.
Come to think of it, this metric is not even continuous, let alone differentiable.
Exactly. Right. And we're using, gradient based optimization. And so what was happening was our experiments were converging within 2 seconds, because of the derivative being 0. Which is not shocking because the metric is discrete in nature, right? So what we actually did was we applied a smooth approximation of the Jaccard metric to kind of help with the optimization. So we kind of looked at Jaccard as we decomposed the Jaccard metric. It's in the appendix to our paper, but we wrote out the Jaccard metric in a particular mathematical form, and it turns out that mathematical form can be written is the limit of a particular function. And it turns out, so what we did was we basically took this limiting function and we used that as our optimization function instead. Just to help us be a little less discrete in the kind of what I call like a step function. We tried to make it a little less of a step function kind of thing. And so that did help with optimization. And yeah, I mean, the parameter estimates, going back to your original question, I mean, the parameter estimates did differ. I think most importantly, I think the parameter estimates, like, I think, when we predict on Jaccard or fit on Jaccard, I think more importantly, we're focused on the predictions based on Jaccard. So, I don't know, I think when you're trying to understand, let's say you want to understand the profiles, right? And how they vary across the IHO positions. then using L2 probably makes more sense because you're interested in the entire composition of the amino acids, right? But if you're only concerned in kind of the high frequency basis as let's say antibody engineer, then using Jacquard is maybe more appropriate.
Well, that's a lot of information to process for our listeners. Before we end, do you have anything else you'd like to mention to plug?
I don't have anything about the paper, but
just in general, whatever you want our listeners to know.
Well, I appreciate that you make this podcast. It's been fun to participate.
Yeah, and just, in case people are interested, we do have a GitHub page for this. We have a command line tool that can predict, these amino acid profiles, substitution profiles based on a single input sequence. And we provide the link in a GitHub link. So if you're interested in that, you should definitely take a look.
Yeah, I'll put those links in the show notes for the episode so the listeners can find them there. Cool, guys. Well, thanks a lot for your time. It was a lot of fun.
Yeah, it was a great chance to discuss this. Yeah.
Yeah, thanks for setting up.